Let's see if you're ready for the MCAT. Which of the following DNA mutations would always lead to a truncated protein if observed in the portion of the coding strand shown below? So the keyword here is truncated. When we're talking about truncated proteins being coded, we're referring to an early stop codon due to some mutation. Now, our three stop codons are UGA, UAA, and UAG. How do you remember this? You go away, you are away, and you are gone. So we're trying to find which of these mutations leads to something like this. Now we have DNA, right? This codon stuff comes from mRNA. However, this is the coding strand, and essentially the mRNA is going to be synthesized by the complement of this, meaning that it's going to look identical to this, our mRNA, except that the T's are all going to become U's. So keep that in mind when we're looking at this stuff. So let's look at all these answer choices. The first one, substituting the second nucleotide for a thymine. If we do that, it's saying that we're going to have 5 T, T, G, T, and whatever, right? Now, looking at this to begin with, we don't see any, stop codons other than right here, this TAG. But if we just substitute a T right here, that's not going to change anything else. We're looking at this specifically as well, right? TTG is not a stop codon, so nothing else is going to change. So A is not going to be our answer. Now, deletion of the fifth nucleotide, deletion of G. You guys can take a look at it, but just to save time, I'm going to tell you that it's not going to do that, either. So this is not going to be your answer. Now, addition of 3 cytosines after the third nucleotide. So if we add it after the third one, right? This is one set of codons. This is another codon. This is another codon. So if we're adding it after the third nucleotide, all we're doing is moving it, right? We're adding another codon in the middle, but that's just going to extend the length of the protein. That's not going to truncate it, right? Because our stop codon is right here, and that's just going to be pushed back. But there's no sort of change to any of these codons that will lead to a truncation early. So this is not our answer. Now, process of elimination gives us D, but let's check this. Insertion of adenine after the second nucleotide. So after the second nucleotide, right? So instead of TGG, we're going to have TGA and then G and whatever. But this is our first codon. And remember this, UGA, you go away. This is going to lead to a truncated protein because it's going to stop right in the beginning. So D is our answer.